we forget about the history of drug development and of our diseases. And it's interesting. The young crowd, I'm not sure how I'm defining young, maybe under 40 or 50, just think these biologics magically appeared. There, there was a lot of science that went into them. So I actually started my presentation talking about the history of Belimumab and BLISS. And it goes back to the 1990s with mapping of the human genome. And they identified a sequence in neutrophils that they actually called neutropene alpha. So they called this sequence neutropene alpha. It was part of the TNF family. And its product, what it encoded, was affecting B cells in a major way. It was a growth factor, a survival factor for B cells. So the company quickly made an antibody to what was called BLISS. So they changed the name from neutropene alpha to B lymphocyte stimulator. And so they made the antibody, which we now know as Belimumab. And so the early studies were not just in lupus, they were in rheumatoid arthritis as well. And even a more interesting piece of information, when we were doing the phase 1 studies with Belimumab in lupus, we were studying BLISS itself in patients with what's called common variable immunodeficiency. So where patients are unable to make antibodies, protective antibodies, and have a lot of difficulty with infections. So the idea was that BLISS could stimulate their B cells to make antibodies and protect them against infections. So in the freezer, we had BLISS, and in our refrigerator, we had anti-BLISS or Belimumab. But what went forward was obviously anti-BLISS or Belimumab. And we did the phase 1 study, phase 2 study, phase 3 study. And so as everybody knows, Belimumab got an approval in March of 2011 for systemic lupus. But there were some signals in the very large BLISS program in SLE that it might have an effect on kidneys, a favorable effect on kidneys. And that was published. So that in part was some of the impetus to do the BLISS-LN trial, BLISS lupus nephritis trial. And that was the largest trial in lupus nephritis ever performed with about 444 patients. And there were a lot of unique features about that trial, which will figure into this presentation that I gave last week. So number one, most lupus nephritis trials only allow patients with proliferative disease into the trial. This particular study not only allowed patients with proliferative disease or a combination of proliferative and membranous, but they allowed patients with pure membranous nephritis into the trial. And that's a liability. Pure membranous, also known as class 5 nephritis, I would say is our nemesis because it's very difficult to treat, or I should say it's difficult to see a response in many cases. And so it's a liability including those patients into a trial because it may dampen your response rates. But anyway, be that as it may, about a little over 15% of the population had pure class 5. Another unique feature was the allowance of patients to come in on background cyclophosphamide. Most of our studies are very strict. They allow patients just to come in on mycophenolate as background therapy. But in this study, you could be on either mycophenolate or cyclophosphamide. And it was around a quarter of the patients who came in on background cyclophosphamide. There were some other key features. For example, at the end of the study, patients had to achieve the endpoint twice. Because these studies can be a little bit fickle, though with lupus nephritis, we're measuring numbers, but you can see variation in numbers. So we wanted to make sure a responder was really a responder. So they had to achieve the outcome measure at 2 consecutive visits at the end. Also built into the study was the requirement to taper prednisone to under 10 milligrams by 6 months. And that figured into the response. So if you weren't able to do that, then you were considered a non-responder. So there were a lot of things that made the BLISS-LN trial a pretty rigorous trial with a hard to achieve endpoint. All right, so then the data came out and the response rate was good and the effect size was good and statistically significant. So we saw an approval of Belimumab, or I should say an extension of the label with FDA approval for lupus nephritis in December of 2020. All right, then about a month later, the voclosporin data came out in lupus nephritis. So now all of a sudden, people are starting to compare trials. And you really shouldn't be comparing results. Trials have different entry criteria, they have different definitions. I could go on and on about the differences. But no one can resist the temptation to compare trial results. Oh, is this drug better than the other drug? And anyway, so then along came the obinutuzumab data. And now people are comparing all three. And so part of the reason for doing this post hoc analysis that I presented last week was to bring the Belimumab BLISS-LN trial on sort of a level playing field with the Regency trial. So just to go over differences in trial designs. So for example, BLISS-LN allowed patients with pure class 5, Regency did not. Background therapies, BLISS-LN allowed cyclophosphamide and Regency did not. The endpoints are being measured at different times. So BLISS-LN measured it at 2 years, week 104, and Regency at week 76, a year and a half. And then the complete renal response definitions can even vary. So we can talk about those, the differences. So in BLISS-LN, complete renal response, the eGFR that we spoke about before could not go up by more than 10% of the pre-flare value or the baseline value. So 10% is not a lot of wiggle room. And in fact, it had to be greater than 90 mLs per minute. So the fact of the matter is that's a pretty rigorous endpoint definition. In Regency, there's a little more wiggle room. 15% of baseline was the threshold, 15% of the baseline eGFR. All right, as far as the reduction in proteinuria, both were the same, down to a protein creatinine ratio on the timed urine of less than 0.5. And that's become somewhat standard. And then as far as sort of allowances for intercurrent event, I mean, both were fairly strict. You couldn't be rescued with other medicines. But as I mentioned, for BLISS-LN, the prednisone had to be tapered to 10 milligrams or less by 6 months. So that's just an example of the differences between BLISS-LN and Regency. There's some other differences, and you could go through every permutation, the Aurora trial with voclosporin versus Regency, or Aurora versus BLISS-LN. But the bottom line, if you start comparing trials, you could compare the designs, but be really careful comparing trial results. Okay, so just to go over the entry criteria for BLISS-LN. So as I mentioned before, pure proliferative, proliferative plus membranous, or pure membranous. And background therapy, mycophenolate, or cyclophosphamide. So again, to equalize the playing field to Regency, the pure 5s and the patients on background cyclophosphamide were eliminated from this post hoc analysis. So we're just looking at what we call the MMF subgroup, the mycophenolate subgroup, and those patients who had either pure proliferative or proliferative plus membranous. And after the elimination of those patients that I told you about, we were left with about 60% of the original cohort. Anyway, so the MMF subgroup had baseline characteristics that weren't a whole lot different than the overall population. So they were pretty equal. And then it's just a matter of looking at numbers. And I mean, I could go through a lot of numbers. It's probably best off looking at the abstract, but I'd say the most important comparison would be the MMF subgroup to the overall BLISS population. Though we did look at the response rates in the pure proliferatives, we looked at the response rates in the proliferatives plus membranous, but I think for the sake of this presentation right now, let's just compare the entire MMF subgroup. So that includes pure proliferatives plus the proliferatives with membranous to the overall population. And so you can look at response rates and you can look at the treatment differences. And you can look at 2 different time points. So I think for simplicity, let's just talk about the week 104 data. All right, so in the MMF subgroup, the Belimumab response rate was 32.6%, whereas in the original cohort, it was 30%. So that difference of 2.6 percentage points may not sound like a lot, but I must say in our lupus trials, every percentage point counts. Now, something that I didn't expect to see is that the placebo response rate actually went down a little bit. So that meant overall that the treatment effect, the effect size, actually increased from the overall treatment effect size of 10.3% to the MMF subgroup having a treatment difference or effect size of 14.9%. So that difference is, 4.6%. It's actually a 45% relative difference. That's a big difference. So the bottom line is when you just look at, I would say, a more responsive, less challenging subgroup, the response rates improve. Now, what about safety? Well, the safety was pretty much the same in the MMF subgroup as the BLISS-LN. I mean, it was the Belimumab safety profile is excellent. In fact, we saw perhaps a greater effect size in safety as far as serious adverse events. Anyway, so safety was not an issue. And so the bottom line is, the lessons learned from this, you have to be really careful. In fact, you shouldn't even be doing it, comparing trial results, one study to another. I mean, everybody does it, but you have to be cognizant of all the different trial design issues. So that was the message. And, if you go back not that long ago, we didn't have any of these new medicines for lupus or lupus nephritis. I mean, we started clinical trials in lupus and lupus nephritis back around 1995. And we had failure after failure after failure. And in fact, Belimumab for SLE was a historical event because it was the first drug for lupus that got FDA approval through the route of a randomized controlled trial. And then, of course, you had the BLISS-LN leading to the approval of Belimumab for lupus nephritis. And that was history making as well, because that was the first time a drug got an approval for lupus nephritis. So now we have a few drugs, and the big question is which drug, which patient, and we need to do a lot of work there. We need to figure out predictive biomarkers so we can match up the drug with the patient. And that's very challenging. So we've come a long way in lupus nephritis and in SLE. And the bottom line is the outcomes for our patients have improved, and no doubt will improve even more. And there'll be more drugs approved for SLE and lupus nephritis. I'm hoping obinutuzumab gets FDA approval, and we'll have yet more choices to make.